This program is brought to you by Emory University.
Good morning. Today's speaker is Dr. Alicia Lyle. She completed her PhD in pharmacology at Emory in 2009 and has done postdoctoral training in Kathy Grinley and Bob Taylor's lab, and now has her own lab and is a funded investigator and assistant professor in our division. Her lab focuses on the involvement of inflammation and oxidative stress in neovascularization and cell proliferation. She'll be speaking on that today.
So I appreciate the opportunity to share what my lab's been studying with you guys this morning. My understanding is that this is possibly a little bit more basic than what you guys typically have here at Grand Rounds, but hopefully I can convince you that Osteopontin is both an interesting target and a potential point at which we can possibly manipulate its expression to help develop some new non-surgical therapeutics for patients with peripheral artery disease. So I'm going to start out with a little bit of overview. I've kept it pretty brief because I think most of it is, stuff that this audience already knows. But essentially, ischemia is defined as the loss of or the insufficient supply of blood to a tissue or organ, and ultimately, it's commonly associated with several cardiovascular disease pathologies, including heart attack, stroke, and peripheral artery disease. And the risk factors for ischemia are your usual suspects, including being over the age of 60, smoking, high cholesterol, hypertension, and then one of the things that most commonly causes ischemia to those tissues is a buildup of plaque in the artery walls or atherosclerosis that leads to a narrowing of the artery and essentially cuts off blood supply to the downstream tissues. And so a lot of the current therapeutic treatments for PAD, heart attack, and stroke tend to be surgical interventions. And so one of the things that we're interested in is identifying potential new targets for therapeutic intervention and non-surgical treatments for these patients. And so the reason that this is relevant is that we know that the body's endogenous response to ischemia is to develop collaterals. And the body does a pretty decent job of developing its own collaterals. And what we know is that when they do this, they're able to bypass sites of occlusion ultimately, either through angiogenesis or arteriogenesis or other mechanisms, to restore blood flow and to preserve tissue function downstream. The problem is that a lot of patients with comorbidities tend to often have suboptimal or impaired ability to collateralize. And so a lot of times you'll see patients with diabetes and other common issues that tend to be poor collateralizers. And so it's really important for a patient to be able to develop collaterals because, as you can see here, when a patient is able to collateralize well and to bypass those sites of occlusion, you ultimately end up with a decrease in long-term cardiac mortality. And so what we're interested in is how those processes happen and how can we make that process more optimal for some of these patients that have that impaired ability. So what processes are necessary for collateral vessel formation? There are a couple that I'm going to take you through sort of piece by piece. So the first one is vasculogenesis. And the way that we define vasculogenesis in this case is basically the honing of endothelial precursor cells to sites of ischemia. They then differentiate and develop into capillary plexuses. And those capillary plexuses ultimately mature to form functional capillary networks. One of the other more commonly known mechanisms is the process of angiogenesis, which is the budding of new vessels off of existing vessels. And this is followed by subsequent arterialization with smooth muscle cells that allows for the increase in blood flow to the downstream ischemic tissues. And then finally, there's also the process of arteriogenesis, which is the enlarging of existing small arterioles into larger conductance arteries. And that process is commonly mediated by changes in shear stress. It also involves an infiltration of macrophages. There's a great deal of matrix remodeling and then subsequent vascular smooth muscle cell growth and proliferation and migration in order to be able to expand those vessels. And so what our lab hopes is that by better understanding the underlying mechanisms that drive collateral formation, we can identify new therapeutic targets for potential pharmacologic intervention. And so one of the things that my lab focuses on is an inflammatory cytokine known as osteopontin. So as the name implies, it was first identified for a role in basically bone formation and regulation in that process. First identified in osteoblasts. Its gene name is secreted phosphoprotein 1, or SPP1, and it's a small integrin-binding ligand in linked glycoprotein, or it belongs to the sibling family. And so as that implies, it's both secreted, heavily phosphorylated, and then also glycosylated. And so typically it gets defined as a matricellular protein rather than a matrix protein because it tends to have a little bit less tertiary and quaternary structure like your traditional matrix proteins like collagen or fibronectin, some things like that are a little bit more organized in terms of their folding. And it's commonly upregulated when some other inflammatory cytokines go up in response to ischemia. To give you an idea of how osteopontin signals, it has an internal RGD motif that allows it to signal through integrin receptors. It also has a CD44 binding domain that allows it to bind to CD44 receptors. It has 5 predicted glycosylation sites, 36 predicted phosphorylation sites, and it can also be cleaved by thrombin and MMPs. So it's a really complex protein that, like I said, is heavily modified by post-translational modification. And so one of the models that we use in my lab to study the role of different proteins in the neovascularization process is the murine model of hind limb ischemia, in which we basically, this is a mouse hind limb. This end is towards the head, this end is towards the foot, and typically what we do is we ligate the superficial femoral artery, both proximally and distally. We then excise the superficial femoral and cut the deep femoral such that you get a pretty severe acute ischemic insult. And what you're looking at on the right-hand side of your screen is a laser Doppler perfusion image of blood flow, and you can see that in the non-ischemic limb, you can see nice blood flow, red being higher perfusion, blue being low perfusion, nice blood flow through the superficial femoral of the non-ischemic limb, but that we've clearly cut off blood flow to the ischemic limb, and the foot's quite blue. So then the question becomes, can we test our potential interventions in this model to see if we can increase perfusion and increase vessel formation? And this is the model that we use. So what's known about osteopontin and collateral vessel formation is that essentially what the Taylor lab showed previously is that when you don't have osteopontin, you're not able to develop functional collaterals. What you're looking at on your left is a wild type in an osteopontin knockout animal. You can see that after hind limb ischemia, the wild type animal after a number of days is able to develop functional collaterals, and you see that increase in perfusion in your ischemic limb. Whereas the osteopontin knockout animals have an increase in perfusion that's significantly blunted because of the lack of osteopontin. And if you look on the right, what you see is a micro-CT angiogram, and these animals have been perfused with a contrast agent. The bone is then decalcified, and what you can see is the vascular network that's left behind. And it allows you to visualize how neovascularization is taking place. There's a lot of angiogenesis, arteriogenesis, et cetera. Allows you to measure connectivity, things like that. And what was shown previously was also that osteopontin knockout mice again have that impaired neovascularization. And so this implies that osteopontin is a critical mediator of this process. Reactive oxygen species are small charged molecules that include superoxide and hydrogen peroxide, and when they're produced at physiologic levels, they regulate a number of cellular processes that include but are not limited to proliferation and differentiation, growth factor signaling, and also cellular migration. But when they're produced in excess of the body's endogenous antioxidant capacities, they ultimately lead to pathophysiologic conditions. And so understanding the role of reactive oxygen species in this process was also something that we were interested in. Reactive oxygen species are second messengers, and as such, their production and removal is tightly regulated. Superoxide can be produced by a number of different methods. It can be generated by a group of enzymes known as NADPH oxidases, can also be generated by the uncoupling of eNOS and also by xanthine oxidases. That superoxide can then either spontaneously dismutate to hydrogen peroxide, or its dismutation to hydrogen peroxide can be promoted at a faster rate by superoxide dismutase, or SOD. Hydrogen peroxide can then be scavenged by either catalase or glutathione peroxidase, which are endogenous systems that are there to then remove superoxide and hydrogen peroxide to neutralize your ROS production. And so again, when you have this happening in excess of, the increase in ROS in excess of your endogenous antioxidant capacity, this is what leads to disease. But what we're interested in is the physiologic levels of ROS and how this is related to the development of collaterals in response to ischemia. And so as second messengers, reactive oxygen species are important effectors of the inflammatory response, and they're upstream of the expression of a number of inflammatory factors. Reactive oxygen species, again, promote proliferation and migration of vascular smooth muscle cells and endothelial cells. They're responsible for the activation of MMPs and also increased tube formation in endothelial cells. And all of these processes are linked to or play some role in neovascularization. And what we know is that mice with less reactive oxygen species production, specifically in GP91 FOX knockout mice, they have impaired collateral formation after hind limb ischemia, suggesting again that ROS are important for that neovascularization process as well. And so one of the questions that we wanted to ask was what happens to osteopontin expression in response to ischemia? I showed you previously that osteopontin, or the lack thereof, you get impaired collateral formation. But what happens to expression in response to ischemia? And so using our MORY model of hind limb ischemia, what we found was that osteopontin expression in the ischemic hind limb, and more specifically in the adductor muscle, so less HIF1 alpha mediated increases and more potentially other mechanisms of increase. We found that osteopontin expression is increased by 20 to 30 fold in response to ischemia, which is a substantial amount given that a lot of other inflammatory cytokines are upregulated to a much lesser extent. And we also see that its expression is increased at the protein level, both in the full-length post-translationally modified form and then also at the cleaved form. And then one of the things that we wanted to investigate was what cell types are responsible for this increase in ischemia-induced osteopontin expression. And so to investigate this, we did some immunocytochemistry. And what you see in these fields is osteopontin will always appear as green. Hopefully this projects okay. And then any other cell marker is in green, in red, colocalization will appear as yellow. And so what you can see is that osteopontin is made by macrophages, by endothelial cells, and then also by vascular smooth muscle cells. And so one of the things that we next wanted to investigate were what are the mechanisms by which osteopontin expression is regulated in response to ischemia, and was there a role for reactive oxygen species? And then more importantly, what specific reactive oxygen species? So we weren't able to detect any significant increases in superoxide production in response to ischemia, but what we were able to detect was a significant increase in hydrogen peroxide production in the tissue when we used an Amplex red assay. And what you can see is that hydrogen peroxide production is increased as early as 3 days, but seems to peak in production around 5 days and then starts to taper back off. And so by observing that there's an increase in hydrogen peroxide, we hypothesized that it was hydrogen peroxide that was regulating osteopontin expression and thus collateral vessel formation in the ischemic hind limb. And so to further test this, we used a transgenic mouse model that overexpresses catalase, more specifically in its smooth muscle cells. Like I said, there are a number of cell types that contribute to osteopontin, and so our primary interest in this case was how much of that ROS from vascular smooth muscle cells contributes to osteopontin expression and thus collateral formation. And so these particular animals express express human catalase, and when you cross them with a smooth muscle myosin heavy chain Cre-expressing mouse, the Cre then excises the GFP reporter and allows for the overexpression of catalase, specifically in smooth muscle cells. And so using these mice, we again used our hind limb ischemia model and saw that hydrogen peroxide in our wild-type animals is significantly increased in response to ischemia, which is what we saw previously, but that when you have overexpression of catalase, specifically in your smooth muscle cells, a substantial portion of that increased hydrogen peroxide in response to ischemia is blunted, suggesting that one, smooth muscle cells are an important source of hydrogen peroxide in this model, and two, that we can decrease it with the overexpression of catalase. And so what happens to osteopontin expression in these animals? So we looked at both the mRNA level and at the protein level and saw that when you have this decreased hydrogen peroxide production by smooth muscle cells, the increase in osteopontin that you typically see in response to ischemia is significantly blunted, both at the RNA level as well as at the protein level. Next, we looked at what happens to collateral formation in these animals when you decrease reactive oxygen species production by smooth muscle cells. And again, you're looking at laser Doppler perfusion imaging, and if you focus on the proximal region, you see that wild-type animals, again, have this increased neovascularization, but that the smooth muscle catalase overexpressors, again, exhibit significantly impaired neovascularization, similar to what we see in an osteopontin knockout animal. And then what's included below are the micro-CT angiograms to confirm that the decreased perfusion is also attributable to a decrease in vessel formation. And so one of the questions that we asked, there are a lot of things that are downstream of reactive oxygen species production, including other inflammatory cytokines. So it was important for us to ask how much of this is being mediated by this loss of osteopontin versus the loss of other inflammatory proteins. And so we basically used a rescue model where we did wild-type animals and added a lentivirus that overexpressed GFP or a lentivirus that overexpressed human osteopontin, the full-length version. And again, what you can see is that the catalase animals have significantly impaired neovascularization, but when you add back human osteopontin A, which is your full-length version, you're able to substantially rescue neovascularization, again suggesting that osteopontin is a critical mediator of this process. And so in conclusion to this sort of part of my talk, we know that ischemia-induced osteopontin expression is hydrogen peroxide dependent, and that the presence of osteopontin is critical for post-ischemic neovascularization. But like most things, it's never quite that simple, and so the direction that my lab has been taking recently and what we focus on right now are the human osteopontin isoforms. So unlike rodents, humans have 3 isoforms that are derived from a single mRNA transcript and are the result of alternative splicing of that single mRNA transcript. And so ultimately that alternative splicing leads to the 3 isoforms shown here, osteopontin A, which is your full length, osteopontin B, which lacks exon 5, and osteopontin C, which lacks exon 4. And this is a place where rodents and humans diverge. Rodents, to our knowledge, don't have isoforms. They only express the full-length equivalent of A, whereas humans have multiple isoforms. And so what was known about osteopontin isoform expression in disease, a lot of what we know about the isoforms to date comes from the cancer literature. And I'm not going to go over this in a great amount of detail, but essentially what was seen was that different tumor subtypes express different isoforms or upregulate different isoforms. But ultimately, when you sort of scan across all of these, the 2 isoforms that seem to be most predominantly expressed, whether you're looking at a breast cancer tumor or a non-small cell lung carcinoma, is that you have an increase in osteopontin A, the full length, and osteopontin C. As we all know, tumors need vascularization to grow. And so looking at that literature, it was really interesting to me that you have increases in very specific isoforms. And so we wanted to ask the question of what isoforms are upregulated in cardiovascular disease, and how do those isoforms differentially mediate collateral formation. And so the take home is that basically isoforms exhibit tissue-specific expression patterns and functions, but how each of those isoforms functions was unknown in cardiovascular system. One of the things that we've done recently is look to see what specific isoforms are expressed in peripheral vascular disease. We had tissues that we acquired from a collaborator in vascular surgery department who gave us tissues from patients that were undergoing limb amputation for various reasons. A lot of these patients tend to have critical limb ischemia, which is why they're undergoing amputation. But it was important for us to ask whether or not osteopontin isoforms were indeed were indeed expressed in that ischemic tissue. And so what we see is that at least in the anterior compartment of the lower limb, and we see similar expression levels in the posterior compartment and also in the thigh muscle, is that we have increases in all three isoforms, A, B, and C. And this is the results from 7 different patients. And so knowing that peripheral vascular disease patients upregulate all three isoforms, the goal of our studies to date have been to define the functional differences of these isoforms in collateral formation. And we hypothesize that these isoforms differentially drive collateral formation through modulation of cell migration and cell signaling. And so the data I'm going to present today are the data that we've generated for 2 of our specific aims thus far. Specific aim 1, which is to determine the role of human isoforms on collateral formation in vivo, and specific aim 3, which is to determine the mechanisms by which human isoforms mediate their differential effects on cell migration. So again, the model that we use is the hind limb ischemia model, and to assess the role of the osteopontin isoform effects on collateral formation, we went back to our osteopontin knockout animals. One, because they have, typically in response to ischemia, you have that 20-fold increase in murine osteopontin, and we didn't want to have to compete with that. So by going back to the knockouts, the only osteopontin in the system is what we add back to the system. That's one rationale for the use of the osteopontin knockouts. And then 2, the knockouts have that impaired neovascularization already. So the question became, okay, well, by adding back A, B, or C, can we rescue that process? And so using the hind limb ischemia model in our knockout mice, each animal received treatment with lentivirus. The construct that we use is shown above. What we have is a SUCMV promoter that drives expression of our human osteopontin isoform, either A, B, or C. Each of these is MIC tagged. And then we also have an RSV promoter that drives GFP blastocyst fusion protein. And so we're able to see GFP as a readout of what cells have been transiently transduced. And so we delivered these lentivirus particles by intramuscular injection using a microsyringe to try to keep the amount of volume that we use to deliver our lentivirus to express osteopontin to a minimum. And so we ultimately deliver 1.5 times 10 to the 6 lentivirus particles in a volume of about 20 microliters. So the microsyringe that we use, we're able to deliver 5 microliters of virus across 4 different sites in the ischemic limb. And then we follow out over time to see what happens to neovascularization. One of the things that we needed to first confirm was that by adding osteopontin isoforms back in this manner, were we delivering similar amounts of lentivirus and were we getting similar levels of osteopontin isoform expression across our animals? So to look at this, we did an ELISA, and we developed this ELISA ourselves in the lab because a lot of the ELISAs available for the human osteopontin isoforms don't actually bind all of the isoforms because of the way that the antibodies have been developed for capture. And so what we used in this case was an antibody that we confirmed actually can and will bind all 3 isoforms, and then we used a MIC antibody to detect how much isoform was there and confirmed that in our animals, we're able to deliver and express equal amounts of A, B, and C. So any functional differences that we see are not a product of differences in expression in vivo, but a product of the actual functional consequences of each of these isoforms. So what we saw is that essentially osteopontin knockout animals, again, have significantly impaired neovascularization, and when you add back LVGFP, you do not see any significant differences. However, when you add back osteopontin B, you also don't see any significant differences. When you add back Osteopontin A, which is your full length, you see an increase in neovascularization. But our most surprising result was that Osteopontin C was able to substantially increase neovascularization over the other isoforms. And so this was a surprising result to us because Osteopontin A is your full length and Osteopontin C lacks exon 4. To determine if these increases in perfusion translate to an increase in functional limb use, we basically put our animals after treatment and surgery into running wheel cages. Each animal has access or free access to a running wheel. They're given free access to food and water, kept in a behavioral suite with regular temperature and regular light-dark cycles, and then we follow how much they run for 7 days. The data I'm going to show you, we put animals in the cages 7 days post-hind limb ischemia and treatment, and then allowed the animals to run for 7 days. And what we saw was that osteopontin knockout animals, again, have significantly impaired limb function. They don't run as well as a wild-type animal. And the add-back of GFP, the animals you see the same thing. But when you add back Osteopontin A and Osteopontin C, you're able to see that they significantly result in not only an increase in perfusion, but an increase in limb function and running wheel use. Again, this isn't a readout that we make the animals run. This is how much they want to run because they're feeling better and want to use their limb, and mice love to run. And so we wanted to investigate how much of these functional effects were due to angiogenesis versus arteriogenesis. And so to look at angiogenesis, we did immunocytochemistry staining with lectin and then counted our lectin-positive vessels across all of our groups. And what we saw were no significant differences between isoform-treated animals. And so from this, we were able to conclude that the differential effects of isoforms is not mediated through angiogenesis. So we also asked the question of whether or not this was mediated potentially through arteriogenesis. And so again, we used the same animals and the same sections and stained for smooth muscle alpha actin. We then quantified the number of smooth muscle alpha actin positive vessels and found that there were no differences in the number of smooth muscle alpha actin positive vessels. But when we measured the sizes of these vessels and looked at their frequency distribution over a range of sizes from the sizes that you would typically see for small arterioles, 10 to 200 micrometers, to small arteries and large arteries, Osteopontin A and C both increased the frequency with which vessels fell in that small artery and large artery size range. So while there was no change in the number of smooth muscle alpha actin positive vessels, a lot of the vessels had shifted in size or enlarged, which is a clear outread of arteriogenesis. And so as I alluded to previously, arteriogenesis is mediated by a number of cell types, including macrophages and vascular smooth muscle cells. So we wanted to look and see what happens to macrophage recruitment in these animals, knowing full well from previous studies that osteopontin knockout animals weren't able to recruit macrophages in response to ischemia. What you see is that GFP had no effect on macrophage recruitment, but the osteopontin A and C seem to have an increase in macrophage recruitment compared to osteopontin B. And when we quantified this using quantitative real-time PCR, we were able to see that osteopontin C increased CD68 expression, or the number of copies of CD68, and we used that as a readout for macrophage recruitment. And so one of the things that we had to ask ourselves, we see this increase in macrophages present in the tissue, but how much of this effect is mediated directly by osteopontin or through indirect mechanisms? One, and then two, what happens to, or how are osteopontin isoforms influencing macrophages? So one of the first things that we looked at was whether or not osteopontin isoforms could directly shift macrophage polarization. And so we used the M1 marker INOS as a readout for M1 polarization and arginase as a marker of M2 polarization. And I think what you can appreciate is compared to our positive controls that are able to elicit a distinct shift, we don't see any shift in macrophage polarization in response to our isoforms, nor are there any real differential effects. To determine if they had effects on survival, which osteopontin has been implicated in previously, though nobody's ever looked at how isoforms differentially affect survival or if they differentially affect survival, We used FACS analysis and saw that, first of all, wild-type and osteopontin knockout macrophages don't have any differences in their ability to basically die. When you put them in serum-free conditions, the cells have an increase in Nexum-5 positive staining and Perpitium iodide positive staining, and they're equal in that measure. So the lack of osteopontin doesn't influence the cell's survival. But the question became, if you try to rescue that apoptosis, can you do so by just adding back recombinant human osteopontin isoforms? And so what you can see in the bottom figure is that when we put these animals again in sort of a downward spiral towards apoptosis, when you add back osteopontin A, B, and C, you're able to significantly increase cell survival, with osteopontin C being the more substantial, or the isoform that's able to rescue significantly better than A and B, although how much of that translates to actual functional effects is unknown. So not only do we increase survival with isoforms, we decrease early apoptosis and late apoptosis. So with all three isoforms having similar effects on survival, it didn't tell us where those differential effects in vivo were coming from. And so we wanted to investigate if isoforms could have differential effects on macrophage migration in vitro. And so to investigate that, we used a modified Boyden chamber assay where we loaded cells into the upper chamber, in this case macrophages, and then put our substrate, in that case the human osteopontin isoform, in the bottom chamber and looked at how cells migrated over time in response to each of those agonists. Cells are then fixed after they're allowed to migrate for 2 hours and stained with DAPI. And then we count the number of cells that have migrated in response to those stimuli. What we know is that wild-type macrophages are able to migrate in response to a positive control MCP1 quite well, but that osteopontin knockout cells aren't able to migrate in response to MCP1. But the question remained whether or not we could rescue that knockout cell migration with osteopontin isoforms. And so what you see here is that osteopontin A is able to elicit a really small response, but where we see our differential effects is that osteopontin C is a really potent stimulus for macrophage migration in vitro, suggesting that isoforms have direct effects both in vivo and in vitro, primarily through the recruitment of macrophages, and then subsequently that pro-survival signal that they can elicit after they've recruited them in vivo. One of the other things that we looked at was the effects of osteopontin isoforms on vascular smooth muscle cell migration in vitro, because again, arteriogenesis has that component that's elicited both by macrophages and vascular smooth muscle cells. And what you can see is that the effects are slightly different on this particular cell type, but the one underlying sort of consistent observation is that C continues to be our most potent stimulus for migration, here for vascular smooth muscle cells as well as for the macrophages. And so when you dig a little bit deeper into what is actually going on in the human osteopontin isoforms, how are they different? All three of them have a conserved RGD domain that's not affected by alternative splicing, nor is their CD44 receptor binding domain. Really what you can see if you look deeper into the sequences of exons 4 and 5 that are missing from C and B respectively is that a lot of what is likely taking place is post-translational modification. And so we hypothesized that the differences that one of the mechanisms by which isoforms have differential effects is because of the presence or lack of specific post-translational modification sites or phosphorylation sites. And so to test this, we looked at vascular smooth muscle cell migration in response to PDGF as a positive control, but also in response to osteopontin A, which I showed you previously increases smooth muscle cell migration. But when you treat osteopontin A with alkaline phosphatase to remove all of the phosphorylations, you subsequently lose A's ability to increase vascular smooth muscle cell migration. And when you add heat-inactivated alkaline phosphatase such that you don't have that removal, you're able to still get migration of your cells, suggesting that osteopontin requires phosphorylation to actually increase cell migration. And so in summary, osteopontin expression is significantly increased in response to ischemic injury. Ischemia-induced increases in osteopontin expression are hydrogen peroxide dependent, and osteopontin is required for post-ischemic neovascularization. We see that osteopontin isoforms A and C are able to rescue neovascularization and functional limb use compared to control in osteopontin B-treated animals. And then finally, the osteopontin C increases macrophage cell infiltration in vitro and in vivo, and also is a positive mediator of cell survival. In conclusion, these results demonstrate that human osteopontin isoforms have distinct effects on neovascularization and cell migration, and may represent novel therapeutic targets in patients with peripheral artery disease. And so with that, I'd like to acknowledge my lab, especially Grace, my postdoc, who's done a lot of the work that you've seen today, as well as my collaborators and my funding source. With that, I'm happy to take any questions.
Thank you. At the beginning, you mentioned that there might be pharmacologic targets and that therapies might be developed now. And just would be interested in your thoughts. So osteopontin is helpful in maybe developing new blood vessels, improving collaterals, but it's also involved in cancer. And I know in breast cancer and a number of things, and it's a target to treat cancer. But it seems like, as far as the blood vessel issue, you'd be doing the opposite effect. So how would you envision ultimately targeting that system for a vascular effect?
It's a good question. So one of the things that we know, and it's pretty preliminary data, so we didn't include it today, but what I showed you on that sort of slide that sort of went over a little bit of the cancer literature was that we see primarily that A and C increase vascularization of tumors. But what we know is that A and C seem to be promoting collateralization in the hind limb, but I think where the therapeutic intervention may come into play for collateral formation is that we know from recent studies that osteopontin B may serve as a competitive inhibitor for the positive effects of A and C. So what I showed is that B doesn't promote neovascularization, that we have some preliminary data that supports that B can impair the positive effects of A and C. And you saw that in our peripheral vascular disease patients, you have expression of all three isoforms. And so what we're interested in is how those isoforms interplay. And so where I would envision therapeutic inhibition would be to inhibit osteopontin B so that what endogenous A and C is around can have their positive effects without that inhibitory effect of osteopontin B. So that's something that needs a lot more investigation and to be teased out fully, but that's what our preliminary data suggest thus far. And so I think because patients express all three, targeting B rather than adding A and C would be where we would actually look to intervene, if that makes sense.
And then another question related to the degree of ischemia that would activate this, and thinking more along the lines of exercise and peripheral vascular disease, and what degree of ischemia does it take? Do brief periods of ischemia activate this, or is it, how does that play a role?
Yeah, so I don't think that that's something that we have an answer to at this point. You know, human studies are pretty lacking in this sort of area of interest, and so, you know, the short answer is I don't know, but it's also something we'd really like to look at. We've never tested, at least in vitro, different degrees of hypoxia and its role in isoform expression in smooth muscle cells or any other cell types. And then it's unclear how much expression you would get with varying degrees of ischemia in a patient. I know that osteopontin is significantly upregulated, and you can detect it in the serum of patients with peripheral vascular disease. So it's a strong indicator for that going on. But I don't know what degrees of ischemia are required for the increases.
Other questions? Yes.
Very nice work, Ali. So down the same line, do you know if in subjects, either mice or human, who have some ischemia, if exercise induces osteopontin?
It's a good question, and we actually don't know the answer to that either. I've never looked at osteopontin expression in response to exercise. It would be quite difficult with mice. As you know, they're so very active. And so that would be difficult. And again, like, this is something that hasn't really been looked in patient populations. I mean, a lot of what's known about osteopontin is primarily focused on its global amount of expression, but not necessarily on isoform-specific expression. And so, again, the answer is we don't really know. This is still really early work, and not a lot of people are working on it right now. So, yeah, it's something that needs to be investigated.
This is very exciting stuff. I'm just wondering if there is any movement. Anybody knows whether there's movement toward trying some of this stuff in the clinic, because the I mean, for years, you know, FGF, VEGF, all these things have been squirted into legs and to try to do things like this. And so the question of, is it expressed in ischemic you know, patients with claudication whose main treatment is exercise? I mean,
Right.
that's what works. I mean, that works better than anything else if they can do it.
I mean, I would
And so
hypothesize.
it's fascinating to see if it's expressed.
I would hypothesize that it is expressed, but I mean, that's something that we would need to test, and we would need likely some collaborators to work with. But I think it's not going to be just a question of whether or not osteopontin is expressed, but how much of each isoform do they have. What we see in vitro that's also really interesting, I mean, knowing that diabetics are, you know, classically poor collateralizers, we stimulated some human aortic smooth muscle cells with hyperglycemia, and found that the vascular smooth muscle cells, human vascular smooth muscle cells, typically primarily express A and C. But when you put them under hyperglycemia, they have a shift in expression towards B. And so I think it's going to be really telling what specific isoforms those patients express. Are they still expressing A and C to try to help promote vascularization in the case of exercise? And then those that don't exercise, do they maybe have a shift towards expression of B? So they're all questions that we'd love to answer in the future.
Looking at your color map, and I might be making too much of this, it looked like that in the ischemic leg, that you approximately had a hodgepodge that might suggest you got microvasculature, and then downstream the vessel is reconstituted. Is that reading too much into it?
A little bit, just because the laser Doppler perfusion imaging is, you know, a relative measure of perfusion and blood flow through heat maps. So it's really difficult to interpret what types of vessels are contributing to that, because it only goes so deep into the limb. And so that's really why we use our micro-CT angiograms to better address the question that you're asking, is how much of that is attributable to an increase in small vessels versus an increase or enlargement in large vessels. That's really difficult to sort of conclude from laser Doppler perfusion imaging. Yep.
Do you expect that?
Is the mic?
So when you measured the amount of osteopontin, was that localized released, or was that like systemic and detectable in the serum?
The levels by ELISA in the animals?
Yep.
That's localized release. So because we use Lentivirus to transiently transduce the cells within the muscle tissue, we primarily looked there because that's where we thought that they would be exerting their primary effects. But as a secreted protein, osteopontin can make its way into the bloodstream. In fact, a lot of the things that have looked at osteopontin expression in patients tend to look in serum rather than tissue expression, because it's easier to get serum than tissue from human patients.
And those parallel findings that you found? The serum expression?
We actually didn't look at serum expression in those animals.
Okay.
I have a follow-up question, Ali. It was very interesting you mentioned the hyperglycemia and the change in osteopontin A and C towards B. And since you said these are different splices of the same mRNA, do you know what regulates the splicing?
It's a great question. Unfortunately, there's not a lot that's understood at this point about what allows for shifts in differential splicing of proteins in general. I mean, there's some data out there. I think ultimately it would be really interesting to see if, what little is known about that, if whether or not we pull down different isoforms, if there are different splice protein regulators bound to the mRNA, but that's something that is pretty far off in the future for us. It's definitely not our area of expertise, but right now what we're interested in asking is rather than, sort of how that splicing occurs is whether or not the splicing is different in response to different agonists. So, we saw that hyperglycemia tips the scales towards B, and that's really relevant to what we think about with respect to collateralization in diabetics, but we actually don't know whether or not hydrogen peroxide tips the scales towards B or angiotensin II, all of which are known to upregulate osteopontin. And so that's sort of the first question that we're asking is whether or not that's a hyperglycemia specific event, or if there's always a shift in B, shift towards B in response to agonists that are known to have bad effects on osteopontin expression.
One other question. There was a slide, looked like a scanning EM or something, some endothelial cells, and it looked like whatever the osteopontin was along, or at least whatever the staining was along the gaps between the endothelial cells. I mean, is that so I'm sort of interested in that because endothelial regrowth is different with a lot of different things, and the gaps may be important for instance, after stenting with different type things, bioabsorbable and so forth, there's a difference in the gaps, and so neoatherosclerosis is a thing we worry about. I'm just curious about whether the concentration is in those gaps between endothelial cells.
It's a good question. I don't actually know the answer to that, nor do we really know the impacts of isoforms on endothelial cell function at this point. But one of the ways that my lab hopes to sort of be able to address and ask some of those questions is through the development of our humanized mice. So we're actually taking osteopontin knockout animals and using CRISPR-Cas9 to put in our osteopontin isoforms, either A, B, or C. And so what we hope to look at in the future is how the isoforms play a role both physiologically but also pathophysiologically, because we really don't know the answer to that. So it'll be really interesting to see how some of these functional effects are altered in the presence of just one isoform or another. But we don't know.
All right, well, thank you very much. Yes.
The preceding program is copyrighted by Emory University.